We're now going to start our session number 3, improving the diagnosis of treatment of CF lung infections and maintaining lung health. We will hear presentations from Professor Andres Floto, University of Cambridge, Professor Jane Davies from Imperial College London, Professor Alex Horsley from University of Manchester, and Professor Jo Fothergill, University of Liverpool, and Professor Alan Smyth on behalf of the CF START trial team. Now, Professor Floto can't be with us today, but he has prepared a talk that we will be listening to. And unfortunately, Jo Fothergill can't be with us today either, but she will be joining live, so hopefully our IT will deal with that. She will be joining us for questions along with everybody else afterwards. So just a reminder, if you have questions for any of the speakers, please put them in the Slido app. We've already got some questions. And James Chalmers will also be joining us for the Q&A after this session. So I think it's just for me now to pass over to Andreas Floto's talk.
My name's Andres Floto from the University of Cambridge. It's my great pleasure to describe to you the Translational Innovation Hub network funded by LifeArc and the Cystic Fibrosis Trust. So this is a joint investment between the CF Trust and LifeArc of £15 million over 5 years to build 4 innovation hubs, which were inspired by and builds on the success of the first innovation hub based at the University of Cambridge, which I was fortunate enough to lead, and which many of you may have heard me talk about previously. But the aims of the network are to fast-track discovery and development of new diagnostics, treatments, medical devices, and other digital solutions to manage lung health and infections in cystic fibrosis. So there are 4 innovation hubs led from the universities of Cambridge, Liverpool, Manchester, and Imperial College in London. And you'll hear from each of the innovation hubs over the course of this session. The idea is to build a collaborative network which brings in partners from academia, industry, and most importantly, people living with CF and their families. And the idea is to be able to overcome the barriers and by so doing, address new ways to predict, prevent, diagnose, treat, and manage CF lung infections, exacerbations, and lung health. So these are the Translational Innovation Hub networks: Trailfinder CF, Flare CF, Pulse CF, and Precision CF. I'd like to just focus in a little bit about the innovation hub based in Cambridge, which is called Flare CF, and it is a collaboration between the University of Cambridge, the Wellcome Sanger Institute, and the Cambridge Centre for AI in Medicine. But the Innovation Hub 2.0 is really focused on addressing 2 critically important questions. The first is, can we forecast and detect acute pulmonary exacerbations in CF? And so what we've shown, I think convincingly, is that home monitoring information combined with machine learning can allow us to predict the start of exacerbations much earlier than we do at the moment. And in a study we ran a few years ago called SmartCare CF, we asked participants to collect sputum samples on a daily basis through a 6-month period. And using our machine learning algorithms, we're able to identify the exact starting point of the exacerbation and to use this information to now start looking at sputum, blood, and breath analysis to detect signals that might help us develop a rapid test to detect a future deterioration in lung health. The second part of the Innovation Hub is really to predict the optimum antibiotic combination. And we're going to do this in a number of different ways. So we aim to deliver actionable intelligence about particular common antibiotic combinations, which could potentially immediately change clinical practice, to predict the response to treatment and the behavior of bacteria in the lungs, and to systematically analyze combination antibiotics, particularly for Mycobacterium abscessus infections. So I really want to focus on the second one of these and share with you some of the recent advances we've made since the start of the Innovation Hub. So over the past 20 years or so, my group has been studying how bacteria cause disease and how to stop them. And what the pattern has been is that we focused on a particular bacteria of importance and drill down and try to understand a key aspect of that infection. Quite depressingly, we realized that there's never enough data on a particular bacteria to create a kind of global, what we call systems level understanding of how that bacteria behaves. And with that in mind, we decided to set out to see if we could create a foundation model to understand bacterial function and evolution at a systems level. And we call this model Bacforma, which is a conflation of bacteria and transformer, and is based on the architecture of large language models such as ChatGPT. If you want to develop a large language model, then you need a lot of input data. And in the case of ChatGPT and others, you scrape the entire internet for text. You need to tokenize these. That is to say, you need to assign a token to each word or part of a word. And then you need to generate informative embeddings. These are higher dimensional representations of these tokens, and train the model using a transformer architecture to predict the next series of words in a masked text. So, for example, the cat sat on the mat. And if you do this enough and at scale, you end up with a number of emergent properties. So say you haven't trained them explicitly, the model has just learnt these. And in the case of LLMs, you could imagine that they have causal reasoning. So say you can have a conversation with them. So if you want to try this at home, then in order to create your bacterial large language model, what you need is an amazing team of computational biologists and machine learning scientists, which we were very fortunate enough to have. And Marcek left a very high-profile AI company to come and work as a PhD student in my lab with Aaron, who's an expert at bacterial genomics, with some great collaborators from Switzerland. You also need a very big computer, and we were fortunate enough to have access to the UK's largest supercomputer at the time, now the second largest, called Dawn, which is based in Cambridge. And because these things were incredibly expensive, we were able to leverage lots of additional funding from the University of Cambridge and the EPSRC. In order to generate our large language model, Bacforma, we needed a lot of input data. And we were able to curate a large corpus of data encompassing over 1.3 million genomes of bacteria. And this represented about 25,000 different species of bacteria living in 70 different biomes or environments. And that was important because we thought that we could leverage information from one species to another species. So the more species we had, the better. And then we needed to decide how to tokenize this information, and we chose this model. So for each bacteria, we looked at the DNA sequence and represented each of the genomes as an ordered series of proteins or tokens. So the tokens were the proteins, and they were in an ordered series as you'd find them in the genome. And so we would take this ordered sequence of proteins, embed them, decorate these embeddings with information around which contig they were in and the exact order in the genome, and put them through a transformer block in order to generate a contextualized protein embedding. And at the same time, we clustered these proteins into families, and the training task was really to predict the family of proteins that was being masked in the genome. So we masked 15% of the genome and trained the model to fill in that 15%. And this worked really well. On the left, you can see what the strain clustering looks like with Bacforma. And each of the colors is a different strain. You can see distinct representations of these in higher dimensional latent space. And without being explicitly taught, the model is able to recapitulate the genetic relationship between families of proteins, genus of proteins, and different species in the embeddings. And in fact, because these embeddings look at not just the core genome, but the accessory genome, it begins to reveal some really important separations of particular species. So in this case, we're looking at E. coli, and the model uncovers 7 distinct groups of E. coli based on their accessory genome, which we think has really important clinical impact. Some of the emergent properties of Backformer, that's to say things that we didn't train but have emerged during training, is the ability to understand the organization of genes into operons, that's to say a set of genes that are regulated by a specific promoter. And in fact, Backformer is really good at predicting these. We generated some ground truth data using long read RNA sequencing, and we showed that Backformer was excellent, as in better than any other model at predicting these operons. It was also able to predict protein-protein interaction. That is to say, it could predict different types of relationships between proteins, such as co-occurrence, the neighborhood, co-expression, using a string database as a ground truth. In fact, it was so good that it was able to predict the protein-protein interactions across the whole genome. And in this case, we're looking at Pseudomonas aeruginosa, and it's showing the interactions between proteins in the whole bacteria. And we were able to test some of these by using AlphaFold 3, which is another AI model, to predict how one protein will bind to another one. And so many of the predictions we were able to demonstrate could have plausible direct binding of the two partners. And really excitingly, it was able to recover known gene networks responsible for virulence in Pseudomonas, which obviously we're really interested in because this is probably the way in which a lot of lung damage is being triggered in cystic fibrosis. We were then able to take this model and train it for specific tasks. So it was really good at predicting gene essentiality, that's to say the genes that must be there in order for the bacteria to live, better than all other models, and protein function prediction. It was really good. And the reason why it's so good at these is because at the protein embedding level, Backformer is much better at embedding proteins based on their underlying function than other models. You can see the colors here are by function. They're much more discreetly separated in Backformer than the best of the rest. And it was also very good at predicting the behavior of specific bacteria. And the way this works, the reason why we looked into it was that we realized that the embeddings of lots of bacteria were clustered by where the bacteria was from, in this case, chicken gut, pig gut, or human gut. It's just an example. But actually, we realized that Backformer was able to accurately predict the behavior of bacteria across a whole range of different phenotypes, from the type of food that the bacteria eats, to its virulence, to how mobile it is, to its metabolism and the shape. And importantly, for our overall aim within the Innovation Hub, it was able to predict the exact resistance to antibiotics of bacteria better than all other models currently available. So this is state-of-the-art performance, which is very exciting. Because of the ability to predict the phenotypes, though, we're able to look at a number of different traits, almost 140 traits across 25,000 species. And this is now allowing us to model the interaction between bacteria in the lung microbiome and the gut microbiome, which are both critically important in CF. It's also an opportunity to use this to discover new genes. And so we were able to recapitulate known genes responsible for bacterial traits, such as sporulation on the left, but also discover genes associated with particular niches that the bacteria lived in. For example, here in Lactobacillus, we discovered genes associated with human gut, chicken gut, and pig gut. And across a separate bacteria, Prevotella, we were able to show proteins responsible or associated with living in particular different animal sites. I'm sure you've all used LLMs, ChatGPT and others, to generate text. And in the same way, we thought, well, let's see if we can generate genomes using Bacforma. And in fact, we can recapitulate the entire genomes of bacteria if we give a few proteins to start off with and set it off. And in fact, you can see here across all protein families, it has pretty good representation. So the predicted here in orange, the true in blue, overlapping pretty much, and across a whole range of different bacterial protein functions. And now we can say, well, hang on a second, let's see if we can generate bacteria with particular properties. So we give it a property token, we start it off with a few tokens, and off it goes, and it generates genomes. And we were able to show that it was able to generate genomes with proteins associated with a particular behavior. So, for example, low oxygen, high oxygen, very cold, very hot. These are kind of test examples. And so to summarize this then, Bacforma represents a foundation model that learns what we call the syntax of bacterial evolution, the proteins responsible for driving particular evolution in bacteria. It demonstrates emergent properties, particularly protein organization and interactions, and delivers the best-in-class performance across a range of downstream tasks, particularly antibiotic resistance. And we can generate template synthetic genomes, which sounds like crazy Frankenstein science, but actually is going to be really important to allow us to use bacteria to create important stuff that we might then develop into medicine. So the next step then for Bacforma 2, we're already on this, is to use more data. And we've got a collection now of about 12 million genomes in collaboration with the European Bioinformatics Institute. And we're going to see if we can tokenize not just at the level of proteins, but also at amino acids and at the DNA level for the bit of DNA between the genes. And we've now got access to the biggest computer in the UK called Isambard AI in Bristol, which is already being used to train this next model. So just to acknowledge the people involved in this little mini project then, so this people from my lab led by Marcek and Aaron, Maria and Ramon in EPFL in Switzerland, and obviously really grateful to the funders, LifeArc and the Cystic Fibrosis Trust, and thank you for your attention.
So thank you very much, Andreas. I think, wow, that's all I can say to that. I think it's going to be a massive help in the future designing antibiotics and understanding infection. So thank you to Andreas. And the next person I'd like to invite up is Jane Davies. And Jane will be talking about precision CF.
It's a great pleasure, particularly as being London-based. I've actually got quite a lot of the local team here today, so thank you guys for coming along. I feel a little mind blown having just watched that stuff from Andreas, so maybe we could just settle our expectations for what might be coming next. We're in the very early stages of this 5-year program, and we're really at the stage where we're recruiting the earliest PhD students, postdocs, etc. So I'm going to tell you what our plans and aspirations are, as opposed to producing data at this stage. And one of the good things about that is some of the plans are still fluid enough that if someone in the room has got the most marvelous idea for doing something different or adding on, we're really open to hearing about them. So whilst we've got good plans, they're not totally set in stone. So our innovation hub is called Precision, and that is polymicrobial infections, focusing on biomarkers and diagnostics, but also understanding mechanisms, particularly of the polymicrobial interactions. The basis of this innovation hub came from the fact that we at NHLI and Imperial have previously had 2 strategic research centres funded by CF Trust around Pseudomonas aeruginosa. In the second of those, we were beginning to look at interactions in polymicrobial states. So in particular, a project on Pseudomonas and Aspergillus, and another one with Pseudomonas and Staph aureus, and beginning to get quite excited and interested at how some of those polymicrobial environments really changed behaviours. At the same time, Darius Armstrong-James, who is a colleague in infectious disease and fungology at Imperial College, had also had a fungus SRC, and it seemed to make sense to perhaps bring the 2 together and see if we could actually focus a little bit more broadly on infection. There are my relationships, most of which have nothing really to do with this particular innovation hub. So where we really began was twofold. And the first was, I'm a pediatric clinician. For many, many years, we have battled with the concept of not being able to get good samples from our children with CF. And now we're actually watching that happen in the adult clinic as well. There are lots of people who previously produced sputum who can no longer produce good quality samples, and that leaves some uncertainty about which particular infections might be in their lower respiratory tract at any one time, whether or not those infections might have responded to a course of treatment. And then on top of that, there's also emerging evidence that mixes of infections may not necessarily always be harmful. Sometimes they could be beneficial. But if we don't know what those mixes are or understand their behaviors, then we can't really predict whether that's a good outcome for the patient or a poorer one. And certainly the first of these, what samples can we look at when we can't produce sputum, was identified as the number 2 priority by the James Lind Alliance. So this is a really big issue now for many, many people with CF. So we built our 2 aims of the Precision Hub around these. So firstly, to further understand the polymicrobial mix and its impact on the health of the person with cystic fibrosis. And then secondly, to try and go beyond sputum and culture to develop accurate diagnostics and biomarkers for detecting infections in people, and to keep those as simple as possible and look at a whole range of these in an agnostic fashion. So this is our team. We are at Imperial and Brompton, but we're also at the university and hospitals in Cardiff, Nottingham, Newcastle and Glasgow. We have some breath scientists in Amsterdam with whom I've collaborated previously as part of the SRC. And we've also for the first time now started collaborating with the CF Foundation's core small animal facility in the University of Alabama, where they have set up rat models of CF infection and are very pleased to be helping us and supporting us with some of our later animal studies of the polymicrobial interventions. We're based around 4 work streams, and what we're doing in these work streams is going from the sort of basic microbes through the microbes interacting with the host in work stream 2, and then into a very large and ambitious work stream 3, which focuses on the person with CF. The whole thing underpinned by work stream 4, which really looks at the education, the dissemination, PPEI, all of those things that are really, really essential in this. So I'm going to walk you through these work streams. So work stream 1 has 4 separate work packages. Luke Alsop's not here today, but he is a non-clinical microbiologist who's an expert in Pseudomonas competition mechanisms and has really drilled down on some of these at a very, very cellular level, and is interested in supporting us look at how these competition mechanisms allow Pseudomonas in particular to get a niche in the airway in the presence of lots of other infections. Similarly, we've got Tracy Palmer, with whom actually we haven't collaborated before, but she's a similar expert in Staph aureus microbiology, and they're going to be working quite closely together, looking at both of their organisms and how they interact with a very large number of other microbes, both pathogens, but also commensals that we think might be quite helpful in the context of the airway. Work package 3 is going to look at trying to establish better models of polymicrobial infection in the laboratory. That's led by myself and Dom Hughes, who's a clinical senior lecturer who's recently joined Imperial Brompton. And then we're supported by colleagues at Imperial who've got a lot of expertise in genome-wide association studies, in metagenomics, metatranscriptomics, and are going to provide us with that skill. Workstream 2 takes this closer to real biology by looking at interaction of these organisms and host cells. So we've got epithelial cells with an increasing array of complexity of epithelial models, through from basic air-liquid interface cultures through to lung on a chip, which Kaz Ito has established at Imperial. We're going to also look at microbes interacting with immune cells, both in Darius's lab and in Glasgow with Robert Gray, who's developed a very big expertise and interest in neutrophil extracellular traps and innate host defenses. and then I mentioned the rat infection model. And this pyramid at the top is really to illustrate that we are trying to do this in an iterative fashion. We're not just going for everything all at once. We're trying to develop some of our early tools, find out which are interesting pathogens which seem to be telling us something really, really important and relevant, and then we will take them through into the more expensive and perhaps the experiments that involve animals. Work stream 3 is being really ably put forward by Gemma Wilson, one of our clinical research fellows at the moment. And this is a very ambitious recruitment across the country of people with cystic fibrosis at all different stages of infection, all different ages and different comorbidities. And we're doing this initially through our own partner centres, but we're hoping to get adopted by CTAP, the Clinical Trials Accelerator platform, and to roll this out and open it to other centres as well. And what we're doing in this is to try and benchmark what we know about that person's infection, and that probably will, at this stage, involve sputum, and it may need to be induced sputum. But then we're going to look at a very large variety of other types of samples, some of them really very novel, like skin secretions, for example, from which we already have an inkling that we can get a metabolic signal that tells us what sort of infection somebody is harbouring. And then my favourite of these various projects down there on the bottom right, the medical detection dogs, which everyone absolutely loves. We can train these dogs to be very exquisitely sensitive and very rapid at detecting a number of infections. So we're actually going to take that forward and see if we can do that on some of these novel sample types as well. And as I mentioned, we're hoping to roll this out nationwide in the relatively near future. This is the sort of thing that we're doing with some of those samples. Julian Fortin can't be here today, unfortunately, but Kath is here, who's going to be doing a PhD with him, further developing some of his home sputum induction models. That's going to really form the basis of our benchmarking of the infection. Group in Nottingham, led by Helen Barr, who you will have heard talk yesterday, has developed a lateral flow system very similar to what we used in COVID, but based on easy to obtain samples such as saliva or urine to look for quorum sensing signals of Pseudomonas that might be a rapid detection system that somebody is harbouring those. We've got breath volatiles, as I mentioned, which is building on the expertise of the Amsterdam group. Darius is looking at systemic omic signatures, and we've got a group based on the volatilomics hub at Imperial College, led by Shard, who's got a lot of expertise in these very advanced mass spectrometers. So you can see we're going in very, very agnostic. What we hope to try and do is to narrow down relatively quickly. I don't think there will be one size fits all, and it may be that one particular technique works best for one type of infection mix and another for something else. This is Alison Evans, who's joined us recently as a project manager. I think she's going to be absolutely key to making sure that this is all underpinned and that we can deliver and that we can get our reports back to CF Trust on time, which I think I did for the first time without asking for an extension this week, and that's thanks to Alison. So thank you very much for that. And you can see these are the various work packages within that infrastructure. And it's largely through some of these that we're hoping to interact with the other innovation hubs. We're hoping that we're going to share our processes. We're going to perhaps share our interaction with PPEI groups. We're going to share our educational offerings so that people within our labs get much broader education across the whole of the network. We are open to collaborations. As I said, we've got a lot going on, but if people have an interest and they think, oh, I've got something that would work well here, we are designing this as a platform so that people can come and plug in and they can test their individual assay, for example, or diagnostic. Once we've got those samples, we'd be very, very open to that. New project ideas or clinical microbes, if people would like to access some of those. This is Amy, who is our technician. And what we hope to have later is that we will get recruitment going before the end of this year. We've just got our proposal in ethics at the moment. And once we do that, we would like to open up opportunities for recruitment more widely, and centres which do contribute to the platform study may actually then have opportunities to apply for some pump priming money to get some of their own projects going, as long as they're in this space. So thank you very much for the opportunity, and it's a real pleasure to be here today.
Thank you, Jane. Now I'd like to invite Alex. Welcome.
Thank you very much. Yes, so thank you very much. So it's my honour to be able to present to you what we're doing in the Pulse CF Innovation Hub, and we've started our clinical study. So I'll talk about that really as that's the main thrust of what we're doing in the first instance, and then the samples and data we generate go into the translational work streams. So Pulse CF, we're based in Manchester, but very key partners in Liverpool and Queens in Belfast, and our focus is on CF exacerbations. And that's important because exacerbations are major events for people with CF. And we've seen that actually at the start of this presentation where patients are talking about their experience of exacerbations. And they're important. Over a quarter of adults require IV antibiotics each year still for exacerbations, and many more on top of that will be having the disruption of exacerbations treated with orals. And they're very important because they contribute to a poorer quality of life, a faster decline in lung function, and carry quite a large healthcare and economic burden. And they're unpredictable and they're disruptive. Our patients live in fear of having exacerbations. That's a quote there from one of our patients. And this is a problem that has been greatly improved by modulators, and all those working in CF will recognize that, but it has not been solved. And James sort of referenced that, I think, at the start, showing this move back towards a less severe inflammatory phenotype, but not back to normal. So we want to get to this point. We want to be able to answer the question, how can we prevent exacerbations? But in order to do that, there is a journey. And the first thing we need to understand is what causes exacerbations in CF. And that's not an easy question to answer because there is no simple single answer. If there was, we would have found it a long time ago. Exacerbations have many parents, and really for every patient, it's not just between patients, but within a patient, each exacerbation is a different event. And it combines the influence of a range of different factors, whether that's the internal environment around inflammation in the lung function, the microbiome, the burden of bacteria, or whether it's external things like exposure to viruses or adherence or pollution. And at the center of that is how that exacerbation comes on and how it responds to treatment. So the hypothesis here is that there are different endotypes of exacerbations that we can identify, and we can use that understanding to predict the severity of symptoms, but also the response to different treatments. Now, a lot of work has happened on exacerbations in the past, but the problem is it all happens at the point when a patient comes to you and says, I feel unwell, I'm having an exacerbation. And because of that, we've got no way of understanding the causes, how we got to that point, and no ability, therefore, to actually, in a focused, evidence-based way, prevent exacerbations. And we also have a single treatment response, so every patient presenting saying they're unwell, no matter how long they've been unwell or what the triggers were. So it's a single hammer for every problem. So if we want to prevent exacerbations, we need to understand the susceptibility factors, we need to understand what triggers off an exacerbation, and we also, as part of that, want to understand who responds to IV antibiotics and how they do so. And that's really where the Innovation Hub comes in. So we have two large clinical studies, both of which are now open, and you know, this has been a huge amount of work over the last 9 months, and full credit to the team who have set that up, but also the teams in the different centres where these studies are now open. And I'll tell you about each of those, so tell you very much more about the clinical stuff here today rather than the back-end translational stuff. So the CF Tracker study is a predominantly community surveillance study to try and understand both the susceptibility factors for exacerbations, but also then what triggers them off. And from that to understand where are the points we can intervene, what are the treatable traits. Unified CF study is for patients who are actively unwell, so coming in for therapy for an exacerbation to try and understand what happens when we give antibiotics, why are some people responding and others not, and what are the biomarkers of response, either to predict what to give or to understand as we're giving things whether they're working or not. And that information will provide what we need to design an evidence-based clinical trial platform for exacerbation prevention therapies. So who's the population at risk? What interventions can we use? What are the outcomes we should be monitoring? And so these are the partners. The main centres are shown there as well, but the academic centres are Manchester, Queens in Belfast, Liverpool, and then the Southampton Clinical Trials Unit will be helping us set up and run the clinical trial. And the idea is that trial will be ready to run at the end of this 5-year cycle. So CF Tracker study, I want to tell you about that because it's important and it's open at the moment. We're recruiting 300 patients across UKCF centres, and it involves a single in-clinic visit lasting around 2 hours where we collect a range of different samples and data. And then the rest of the study is by home monitoring. And we worked hard with patients to make this as light a footprint as possible and as doable as possible. That's why it's a single visit, and then it's every 2 weeks for 6 months. I mean, obviously in an ideal world, we would have done it every day for a year, but that's quite a big ask. So patients will be doing home spirometry. They will be sending back a postage kit, which they can put in the post box, or they can get Royal Mail to pick up. The viral PCR swab there and saliva sample. There's a study app which reminds them when it's time to do this, and also links through to a questionnaire. And then we follow up their data for a full year as well. If they're unwell, they send back additional samples. And then in our core centres, so Liverpool, Leeds, Cardiff, Manchester, Newcastle, 100 of those 300 patients will be having an enhanced follow-up. So they have more assessments and samples collected at the face-to-face visits. They have a potential to do that at 1 month again, and then we repeat that at 6 months. And then in Manchester, we're also putting air quality monitors in the homes to understand indoor air pollution, and with also some more detailed work on outdoor pollution exposures, and also looking at the fungal burden in the air in the home. So what does that look like? So this is essentially multi-domain phenotyping, and behind all this sample collection are a whole range of really exciting translational work streams. We have 5 work streams in total. We have, when we're up to speed, we'll have 10 PhD students. We've got around 4 or 5 already appointed. And we're looking at a range of different things. So we are obviously looking very closely and in a lot of detail at the lung, so the microbiome of the lung function, lung symptoms. We're looking at exhale breaths, we're looking at VOCs, we're also looking at things like NO as an allergic signal. We're looking at the upper airway, so nasal swabs, nasal inflammation, taking saliva samples as well. We're looking at, as you might understand, at the blood, and looking at different cell types, both the makeup of the blood cells, but also their response to challenges, as well as inflammatory biomarkers. We're looking at sweat chloride as a measure of CFTR function. We're also looking at the gut and taking samples to understand the gut microbiome and the lung-gut axis and how that changes with antibiotics. And then we're looking at the external environment, the exposome, so viruses. We're looking at indoor air quality, trying to understand medication adherence and behavior. And this is some of the stuff. It's not a comprehensive list of everything that we're doing, but we are building up as complete a picture we can as what is happening to patients. This is what a home sampling kit looks like. So at the visit, patients go home with this box. It contains within it the home sample kits that they need, as well as additional spares, as well as boxes to use when they're unwell. The study team deal with the home sampling, so if you are a study site, it is really just that first visit that you have to deliver. There are videos, QR linked for the patients so they can remind themselves how they collect samples, and there is a study app, it's on Android and Apple, that sits on their phone, has all the study documents on it, sends them push notifications and reminders and puts that in their diary, and also links out to the CFRSD so they can complete that every 2 weeks. And so far we've had very good returns, so really very few missed sample points. So who's eligible? We've tried to make this as broad as possible. We do need to have patients who are at least have a chance of exacerbating, so it's at least 1 previous exacerbation, either with oral or IVs in the last 12 months. They also have to have a home spirometry device and be able to use that. And importantly, because sputum is such an important medium, they have to be able to produce sputum at visit 1, and that can be through induction. We don't want patients with ABPA or on long-term immune suppressants, unfortunately, because that will muddy the waters. Very briefly tell you about the Unified CF study. So we also want to understand what happens when we give antibiotics. Why are some patients responding, why are and others less so, and what is happening to the microbiome? It was such a good idea that Liverpool came up with a very similar plan as well. So rather than competing, we have a combined protocol across a number of centres now, co-led with Liverpool Hub. So Freddie Frost is my co-chief investigator on that. We'll be looking to recruit 125 CF adults and up to 50 children, which will be in a separate study. We're collecting clinical data at a number of key time points. So the first one is a stable baseline, but importantly, that can be the CF tracker study visit. So if they've done that study, they don't have to do anything more unless they become unwell. We also need to get samples before antibiotics, then repeatedly during retreatment, and then obviously at recovery. We're very interested in looking at antibiotic levels in the sputum and blood. We have capability of doing that in Manchester, but we also want to understand more about the broader impacts of exacerbation, so what the social and economic burden of being unwell if we're proposing exacerbation prevention therapies down the line. So hub progress, I've got to rattle through this because I'm running out of time, but we're up and running in 6 centres. We're setting up in 12 more. We've started very recently, so we've only got relatively small numbers, but we've got around 50 booked through to the end of the year, a number of associate PIs. Unified has literally just opened, got the green light on Monday, and we will be setting that up in more centres, and it will also be opening out to CTAP centres as well. So almost last slide. What's the impact going to be on CF? So we are delivering a systems biology approach to exacerbations. We're doing that phenotyping both at scale in 300 patients, but also at depth in those core centres. This is pathway agnostic. We're collecting a lot of information and working with some excellent existing teams and technologies. And we're creating a resource for CF research, and the ambition is the database will be open access at the end of it. And this is relevant for the majority of people with CF in a post-modulator era, but also very important for other respiratory diseases that we've heard about a little bit overlap with non-CF bronchiectasis. If you want to find out more, it's all on the website. You can follow that QR code for video. Everything's on there. You can find me afterwards. This is flashing because I'm out of time. But thank you very much.
Right, thanks, Alex. So we're now going to move to Jo Fothergill. Hopefully, Jo is going to be online. Hi, Jo.
Hi, hopefully you can hear me.
Yes, we can hear you, and we're looking forward to your presentation. Over to you.
Thank you. So it's my pleasure today to present the Trailfinder CF Hub. I do apologise for not being there in person. I'll acknowledge COVID and my kids for that one. So the Trailfinder CF Hub is a real collaborative effort. And in particular, I just want to give a shout out to Paul McNamara and Freddie Frost for really helping me drive this hub forwards. But you can see all of our partners there. The hub is really thinking about treatments, and that is about supporting new treatments, but also optimizing the ones that we already have access to. So I'm going to give you a brief overview of the two main translational streams, and then a bit of insight into some of the other stuff that we've been doing. So the first stream pertains to advancing phage therapy. So hopefully more and more people are aware of what phage therapy is, but if you haven't heard of it, it's actually a therapy that revolves around bacterial viruses. So these are viruses that specifically infect bacteria and can lead to lysis of the bacteria. So very, very personalized and exciting therapy. We've known about this for a very, very long time. But in the UK, we've been quite slow to take up the potential of phage therapy. And phage therapy has an interesting past with some sort of problematic clinical trials as well. But more recently, this has been quite an exciting area, and that really relates to a government inquiry into the use of phage therapy in the UK. And of course, in the CF space, we've seen BioMX really leading in this space with some exciting data. So I'm going to give you an overview about what we're doing in that space. And like the previous two hubs, we started about sort of 9-10 months ago, but this stream is one that started first. So we've got a number of work packages, and they look at enabling access to phage therapy now for the people who need it, but also filling in some of those key science translational gaps in order to support high quality trials and innovation in the future. So we're establishing a framework to bring compassionate, so named patient use of phage therapy to patients as soon as possible. And we're also, in conjunction with the CF Syndicate and AMR, developing target product profiles for phage therapy in people with CF. We're looking at expanding biobanking and phage libraries, and that's with particular relevance to those highly resistant bacteria that we sometimes see in people with CF. We're looking at developing rationally designed phage cocktails that work in the lung environment, so picking apart those interactions between phage, their bacterial hosts, and of course the wider lung environment. We're looking at refining phage formulation, really drilling down into the pharmacology, particularly around dosing, and looking at delivery routes for clinical trial readiness. Another issue is the lack of GMP manufacture in the UK and very limited GMP manufacture of phage globally. So we're looking at developing that scalable phage manufacture capabilities in the UK and the associated bioanalytics that go with that. And of course, new therapies often need novel diagnostics alongside it. So we're looking at 2 different platforms to test those phage resistance diagnostics. So looking at just a quick overview of some of the progress in that stream, for the central clinical pathway, we've managed to establish a post in UKHSA to be that central contact and coordinate those inquiries and acquisition of phages. We've helped coordinated 5 inquiries from people with CF so far, and we've helped translate and advance the Burkholderia gladioli phages that we've found, and they're progressing through safety and manufacture processes. And that piece of work is really led by Josh Jones, now at UKHSA. In terms of the target product profiles, we're working with the CF Syndicate in AMR, and we've established an expert working group to help us with this. We've performed patient and clinical focus groups and one-to-ones, and we've got an landscape mapping which is underway, and these figures highlight some of the data from that, so you can see the landscape of clinical trials and their phases, and also the types of different niches in which people are currently developing phage therapy. In the biobanking space, this is really being led by Leicester, Cardiff and Exeter. And I think one of our PhD students is in the audience. So this shows a phage cloud. So the dots in blue represent Pseudomonas aeruginosa phages, and the ones in red represent Staphylococcus phages. And you can see that there's limited access to other CF relevant phages. So we've got PhD students expanding the Pseudomonas aeruginosa databases, and Rebecca, I think, has isolated 63. We've got 21 new Achromobacter phages, and we've used a deep genomic mining approach that has resulted in 15 new Burkholderia phages, including ones against Burkholderia gladioli and Burkholderia synespatia for people with CF. In terms of efficacy in the lungs, we've got our PhD students in place, and that work is underway. And these figures just highlight at the top here, we've got efficacy of 6 phages against 900 Pseudomonas clinical isolates. What I really want you to take away is the area in white, and those are isolates in which the phages are poorly infect, so the need for new phages in that space. And at the bottom, you can see a heat map showing what happens after Pseudomonas interacts with some lung components. So the dark blue shows resistance to phages, but you can see it's not universal. So some phages, the bacteria can become resistant to some phages, but not others. And that can inform our cocktail design. In terms of delivery and PK/PD, these figures show some initial work that we're building on, looking at different routes of delivery, so intranasal and IV. And what you can take away from those is that we get good penetration around the body, and regardless of the route of these 2 different administration methods, you can see good amplification of phages in the lungs, which are the first dots in that graph. In the manufacturing space, we're working with a government catapult, CPI, and also the University of Loughborough. And we're looking at developing a GMP plan with a GMP consultant. We're going for MHRA advice on that. We've developed a scalable manufacture up to 5 litres, which equates to around 200,000 doses of phage. And now we're looking at how we can easily translate these to different species so that we can support that named patient basis. And in the diagnostic space, we're trialing 2 different methods. This one just highlights a novel method that could give you a result in 2 hours, and it's based on impedance cytometry. The next stream is about optimizing treatment for acute pulmonary exacerbations. And some of this work you'll see is shared with Manchester, and that's really around the unified CF study at the bottom there. And that's defining those clinically relevant exacerbation endotypes and looking at effective treatments. And that, we're pleased to say, is open, and then the paediatric study will follow. The other questions that we're really interested in this stream are about whether we can use evolutionary microbiology to predict treatment outcomes and mitigate AMR by providing personalized antibiotic approaches. And we're looking at electronic health records to inform those antibiotic choices. And we're also trialing the utility of a approved rapid diagnostic in the exacerbations in CF. And so in terms of progress, this stream has started slightly behind the phage one, but we've got our methodology. Our PhD student started yesterday, but the methodology for the high throughput microbiology is in place. And in terms of the electronic health records, we've got a mathematics postdoc in place who's looking at this data and asking clinically relevant questions like, if one treatment course has been effective the first time round, would it be effective again the second time round? In terms of the diagnostic, we're trialing the bioMerio BioFire Pneumonia Plus panel, and we've trialed that on chronic respiratory samples, and we've got a clinical fellow in place on the implementation work. And in unified CF, as Alex has already gone into details, I won't go into too much detail, but we're taking that multi-omics approach to try and define those exacerbation endotypes, really building on some of the work we've done previously in other clinical trials. Sorry, slight technical issues there. And so the other activity that we're doing in the Trailfinder really is about PPIE, and Jane's highlighted the importance of that. We want to look at inspiring the next generation of work researchers, but also about embedding that patient voice throughout. We've already done quite a lot of work in this space. Particularly, you might recognise a few people in this picture. These pictures: Rebecca Long Bailey, Sir Richard Leese, and a group of us at the Houses of Parliament. We did the British Science Festival about 2 weeks ago, and this gave us the opportunity of working with an augmented reality company. You can see the video at the bottom there, where people could come and make their own phage. It was rapidly turned into a 3D image that you can then capture and put into a game where you can kill bacteria. So really important engagement there. And we're also working on training, and we've got some really exciting training, and we're planning on expanding that out to other hubs, but also to the research community in general. So if you're interested in this, please get in touch. These pictures here are from the Summer Phage Bootcamp, and I've used these ones in particular because this is actually our project manager, Louise, who's never picked up a pipette in her life, isolating new phages for the first time to contribute to our libraries. So this is working on a citizen science type approach, and this has now been developed into an undergraduate practical, so 200 students a year will be contributing to this. So if, like the other hubs, we're really open to collaboration. And if you're interested in any of these, please connect with us on any of these platforms or email. We'd love to hear from you, and we'd love to expand this out to further people as well. So thank you very much for the opportunity, and hopefully that worked okay.
Thank you, Jo. That worked brilliantly. And we'll hope to see you back in the question and answer session. So finally, I'd like to ask Professor Alan Smyth to come and to the podium to give a presentation on CF START, a randomised registry trial.
Thank you, Deborah. And I feel a bit like the poor relative because I'm telling you about a randomised controlled trial which is testing a single hypothesis. And we've heard about some terrifically ambitious programs of research with real discovery science going on. So I hope you'll forgive me for doing something a bit more modest. My other disadvantage here is I'm not Kevin Southern. And Kevin Southern has led assembled and then led a terrific team that has delivered the CF START trial and has recruited 485 infants with CF diagnosed through newborn screening and recruited those into a randomized controlled trial looking at prophylactic anti-staphylococcal antibiotics. And that really is an extraordinary achievement. Kevin can't give this talk today, unfortunately, so I'm kind of a Kevin Southern tribute act. I am the no-oasis to Kevin's oasis, but I am live in 25. But I'm going to take you back a bit to some CF history because really what the CF START trial is doing is it's saying we've been giving this treatment in this way for decades, and really, should we be still doing that? And it's applying that question to the use of prophylactic anti-staphylococcal antibiotics, which in the UK is mainly flucloxacillin, given to very young infants diagnosed by newborn screening. But if we cast our minds back to how we got here, we go back to Dorothy Anderson in the 1930s. She first described CF, but in 1949, she published a paper describing some of the microbiological aspects of the infection. And I've pulled some qualitative quotes from this paper, and I think they are particularly telling today. So she says one of the most characteristic features of the disease remains to be explained, namely the development of persistent bronchitis. This is usually the cause of death, either directly or as a suppurative bronchitis leading to bronchiectasis, or indirectly via cor pulmonalis. And the final quote, the bronchial secretion is viscous and may be abundant, and many of us and our patients have experience of that. And she goes on to talk about the organisms. Cultures taken early in the course of the disease grow Staph aureus hemolyticus in nearly every case. A mixed flora is present in cases with chronic bronchiectasis with Pausanias, that's Pseudomonas aeruginosa, as a common associate of the Staphylococcus. And it's interesting that even in 1949, there was a recognition that these two organisms could be common associates and that one may facilitate the other. In fact, there may be collusion between these organisms. And that kind of underpins one of the questions in CF START, which is, okay, maybe flucloxacillin is helpful in suppressing or preventing infection with Staph aureus, which was clearly feared for decades, but actually could that be at the price of this common associate, Pseudomonas aeruginosa, becoming more prevalent or appearing earlier in the course of the disease? I've put the bottom figures there really just as a sober reminder of how far we've come in CF. So what we're looking at is survival to age 5 in the right-hand column, which in the pre-penicillin era in infants with early onset of cough was 14%. Versus 24% in the post-penicillin era. And the figures are 19% and 36% for late onset of cough. So delayed respiratory symptoms. But I think this is a really sober reminder of how far we've come in CF care, both in terms of well-being and survival. So every good clinical trial starts with a systematic review, and this systematic review has been through many iterations over about 20 years, being updated as new data become available. And I had the privilege of doing the first version of this review with a person with cystic fibrosis, who was also a medical practitioner. The latest version I've done with Margaret Rosenfeld and with Ollie Rayner, who's also a person with CF. What we've found was that there were 4 eligible studies, 400 randomized participants. And if we think about that for a moment, CF START has 485 participants. So a single randomized controlled trial will have more patients than all the previous studies put together. And in this systematic review, we find no significant difference between the groups in infants or conventional lung function. We find no effect on nutrition, hospital admissions, or additional courses of antibiotics. But I guess that's not what we're really interested in. What we're really interested in is staph infection. So I'll take a moment to walk you through this forest plot. The red line in the center is the line of no effect. On what you might call the y-axis, so the 1 to 6 on the left-hand side, you've got the number of years of prophylaxis given. So different studies treating for different lengths of time, up to 6 years of treatment. And where you have a blue box with a line that is an individual study in the 95% confidence interval, where you have a black diamond, that's all the studies for that time period put together, and the big black diamond at the bottom indicates the overall effect. And when the black diamond doesn't overlap the line of no effect, that means there is a significant effect of prophylaxis, in this case on Staph aureus, and where it lies to the left-hand side, it favors prophylaxis. So the long and short of it is that for every time period for 6 years, the use of prophylactic anti-staphylococcal antibiotics means that you get less staph. So it does what it says on the tin. But of course, our fear with this is that by doing this and giving this preventative treatment and possibly preventing staph-related exacerbations and pulmonary damage, actually we could be promoting something else, something worse. And these are the data for Pseudomonas. So here we want to see the black diamond on the no effect line. And mostly it is, but you can see for years 4, 5, and 6, there's a tendency to favor as required treatment. In other words, there's a tendency for prophylaxis maybe to promote Pseudomonas infection. So for that reason, the UK guidelines recommend prophylactic treatment up to 3 years, but no longer. Those guidelines are currently undergoing an update. And ultimately, I think we'll include the CF START data in those guidelines. But you can see why there's caution about the more long-term use of prophylactic anti-staphylococcal antibiotics. And there's a gulf, probably a yawning chasm between US practice where prophylaxis is not recommended, and it's no coincidence that the longest prophylaxis trial in that group I showed you in that systematic review was a US study. They used cefalexin, so they used a cephalosporin prophylaxis, whereas in the UK we use flucloxacillin. But this trial has been very influential in the US and has influenced their practice there, where they don't use prophylaxis. In contrast, in the UK, as I said, prophylaxis is widely used and recommended in guidelines, which takes us to the CF START study design. So I'll walk you through the PICO of this study. The population is newborn screened infants with CF. The intervention, so Kevin came up with these really snappy summaries of intervention and comparator. So prevent and treat is the intervention, comparator is detect and treat. Prevent and treat is you give prophylaxis, you do microbiological surveillance, you treat what you find. The comparator is just treat what you find. So it's a national study. In a moment, I'll show you the number of centers participating, looking at the most effective antibiotic strategy for infants with CF. One of the really defining features of this study is it's registry-based. So the data are collected in a module that sits within the CF registry and allows data to be transferred from the registry to that module, and then additional trial-related information is entered into that module, and the module was also used for randomization. And I think it's worth taking a moment to thank the CF Trust and the registry team for the incredible work that they did to make this happen in CF, and the kind of efficiency and cost effectiveness in trial delivery that has allowed us to leverage. The outcome, paradoxically, for a study of anti-staphylococcal prophylaxis is actually age at first growth of Pseudomonas aeruginosa in a respiratory culture taken as part of a standard visit and entered onto the registry. So the study design looks like this. You can scan on the QR code if you wish, and that will take you to the ISRCTN registered protocol. 480 infants was the sample size, 485 achieved, prevent and treat versus detect and treat, and then the outcomes monitored for 48 months. And that's why I can't present the findings today, because some patients are still in follow-up. I understand from Kevin and the team that there will be a small modification to the follow-up period. We'll be curtailing it slightly. Follow-up will end in May 2026, so May next year. And we hope that we can do database lock cleaning analysis and have some data by the end of next year to inform our practice. A really, really important sub-study is shown at the bottom, and I'm indebted to Molly Riley for providing some photos that I'll show you in a minute, but that's multi-breath washout done with Paul Aurora at Great Ormond Street. Terrific piece of work in a subgroup looking at the effects of prophylaxis or not on lung function at between 3 and 4 years of age. So a huge achievement to open 63 sites across the UK, a huge vote of thanks from me to our collaborators in all those centres who helped recruit those patients into the study. These discussions with newly diagnosed families with a baby with CF take place in the local unit, and this was the place to do this recruitment and randomisation. This is how recruitment, well, let me show you. This is how recruitment should have looked. The blue line, it was all going to be pretty fast. This is how it actually looked. The difference on the x-axis between those two lines is 2 years. So that took some difficult conversations with NIHR Health Technology Assessment about funding, led by Kevin. It also took the faith and commitment of the people in this room who, in spite of the fact that things were going slower than we expected, helped us continue and deliver the study. So what have we learned? I think once recruitment got going, it was steady. I've mentioned our planned full recruitment in May 2023, hope to finish May 2026. The registry worked well, the module worked well, the data quality are good, and we were able to do some additional studies led primarily by AHPs with great enthusiasm, and that was looking at things like breastfeeding. and doing additional tests outside the normal clinical parameters is possible, a little bit challenging, and LCI, lung clearance index, is probably the best measure of this. So thank you to Molly for sending me these slides. Again, I have the privilege of describing somebody else's work here. So this is terrific work by the team at Great Ormond Street. They're studying children aged 3.5 to 4 years. And so far, I think the one on the left is incentivized spirometry. And that's Molly in the middle. Then just in the middle to the right, that's LCI measurement. And on the far right, that's the kind of incentive, the sticker incentive that children are offered to participate. But terrific success rate. So what happens is the families give consent to contact. So this has been done in 186 of those 485. Of those 186, 141 tests have been done, and 107 children have a valid LCI result at 3.5 years of age. So that's a success rate of about 70%. That is a terrific achievement on the part of the GOS team, and some really nice qualitative feedback from families where this has been a great day out. They describe Great Ormond Street as the fun hospital. So that's really all I wanted to say. The follow-up period should end next May. The data should be available hopefully towards the end of next year. I chair the antibiotic guidelines group for the CF Trust, and we're very excited. It may have to be an addendum, but we're very excited about including these data in due course. But I want to pay tribute to Kevin's really important contribution to this and his leadership, and thank everyone who's participated in the study, and thanks everyone for listening.
Alan.